Welcome to this class on Neuroscience of Human Movement. So this is part 7 of our discussion on cerebellum. Right. So in this class we will continue our discussion on spinocerebellum. In the last class we introduced the notion of spinocerebellar modulation of movement. We said that this is modulation rather than control. Right. So in this class we will discuss the organizational principles associated with the spinocerebellar modulation or regulation of movements, and what is the possible role of parallel fibers in motor coordination, because we have 100 billion of these. What could be its role? What could be the role of these large number of neurons, 100 billion neurons? What could be its role in motor coordination and motor function? The following are the three important crucial organizational principles. How are these learnt? Fundamentally, it is important to note how these are learnt. First, these are learned from animal models. So, whenever we say animal models, there are multiple ways in which experiments could be performed on animals. These one obvious way, and an important this lesions on animal models. And the second one is, it is possible to inactivate specific regions of the cerebellum using methods such as cooling. Inactivate specific regions. This may be cerebellar cortex, this may be deep cerebellar nuclei. By this we could simulate a situation in which that particular region or the set of neurons that are affected by this cooling are not functional for the particular period of time, and then observe the effect of this perturbation on the system. Now, obviously lesions are permanent damage or permanent effects, whereas cooling has less permanent or temporary effects, is it not? Obviously. So, from these animal models and studies, we have learnt a few lessons, and these lessons are listed here, and these are first is that Purkinjean neurons and deep cerebellar nuclei, or neurons in the deep cerebellar nuclei, discharge vigorously in relation to voluntary movements. What does that mean? It turns out that when voluntary movements are happening, or just before voluntary movements are happening, it turns out that Purkinjean neurons and deep cerebellar nuclei neurons discharge or produce activity or manifest activity that resembles the activity in the motor cortex in at least two different ways. First is that the delay is similar to motor cortex. So, this is surprising because we said cerebellum does not directly project to spinal motor neurons. We have said this earlier, we will continue saying this. This is a crucial principle. Cerebellum projects to other motor areas which further project to spinal motor neurons and control movements. In that sense, we said, even in the previous class we said that the cerebellum only modulates movements and does not control movements per se. However, here I am saying that the delay of the time delay between the activity of these neurons, the Purkinjean neurons and deep cerebellar neurons, and the action and the voluntary action or the movement activity or the muscle activity, that delay is similar to the delay between M1 neurons and muscle activity. This is about 100 milliseconds, give or take, right, or maybe even a little less. That is surprising because cerebellum must project to some other motor area and then project to the spinal motor neurons and then modulate movements, whereas M1 directly can project to spinal motor neurons or maybe through interneurons, but M1 it is known projects to the spinal cord through the corticospinal tract and through the pyramidal tract, we know these things, right. So, when I say that the delay is similar to M1, then it implies, then we are asking the question, what does this mean, what could this mean? Does that mean that cerebellum is the other cerebrum? Does that mean that the cerebellum performs functions that is similar to motor cortex? The answer is no. It turns out that the cerebellum forms a recurrent loop right. The cerebellum forms, or at least it forms part of a recurrent circuit synchronous with motor cortex to modulate movements. So, it is an important part or a crucial critical part of a recurrent loop in control and modulation of movements. So, it is not controlling movements like the M1 does, but it forms an important element of a recurrent loop in this process in the control of movements. Okay. So, that is one. Two is somatotropy. We have seen this earlier that there is somatotropy in the primary motor cortex. We have seen this, we have discussed this, this is the Penfield's map, we have discussed in great detail to what extent is the primary motor cortex somatotropic, etc. right. It turns out that the deep cerebellar nuclei are also somatotropic, but slightly different from that. The anterior portion contains, or the anterior parts contain the tail and the posterior parts contain the head, in other words it is upside down, right. And also that the medial parts, the medial regions contain the limbs and the lateral regions contain the trunk. So, again it is turned sidewards, right. So, so projections from the Purkinje neurons to the deep cerebellar nuclei is somatotropic. So, there is topographical organization similar to M1. So, once again this raises the question, does this control movements? The answer is that no, once again the same, we have to come, we come to the same conclusion that the cerebellum participates or at least is a critical part of the recurrent loop in movement modulation. So, this is what we learned from the first lesson. This is that Purkinje neurons and deep cerebellar neurons, their discharge is vigorous in relation to voluntary movements, sometimes very comparable to motor cortex, yet we only believe that their role is in modulation of movements. And also then what does it control, what does it modulate? When you say modulation of movements, what does it modulate? At least it is now known that it modulates the direction of movements and it modulates the speed of movements. So, when you say speed, we are talking about some form of movement, vigor, some form of vigor, right, how fast you can move. That is one. The second important lesson that we have learned is that the cerebellum anticipates the movement and the contractions that are required to perform the movement. So, in that sense, if I have to reach a particular point at a particular point in time, the appropriate muscle contractions that are required are anticipated by the cerebellum. How does the cerebellum do this? That is a controversial question that takes us to the second, that takes us to the next lesson. Okay. But first this one. So, the cerebellum is believed to perform this function of anticipation by feed forward control of muscle contractions, thus regulating timing and sequencing of movements. Now, we will discuss some examples in the next few slides. Okay. So, there is anticipatory or feed forward control of muscle contractions, so that appropriately timed sequence of movements are produced. This is crucial, right. At a broad level, at least the following can be said: you do not wear the suit and then go to bath, right. So, there is a sequence in which these things are to be done, but that is at a very broad level. But I am talking about here, I am talking about movements that are performed at a smaller time scale, right. It is not so several and people who have lesions of the cerebellum show typical inability to properly time and sequence movements. So, that also tells us that cerebellum probably participates and contributes in anticipation of movements or proper muscle encoding of these movements that are required. Okay. Then the third is a controversial claim that the cerebellum has internal models, I am putting it in quotes for this. So, in some sense at least the following is known, that when you are making a movement, the limb structure and the dynamics, the mechanics associated with the movement must be properly accounted for to ensure that the movements are smooth. And it is now well known and well accepted that humans are extremely good, extraordinarily good at performing smooth movements. How do we do this? To that extent, it is now posited, at least it is believed by at least some of the scientists, that the cerebellum has internal models. These are models that can do one of the following two things. I will just name these can be either forward models or inverse models. Now, these internal models can either be forward models or inverse models. Now, what do I mean by that? Let us in kinematics, the following is known, that if I know the end point that needs to be reached in a multi-link chain, if you know the end point to be reached, then computation of individual joint angles in that multi-link chain is called as the inverse problem of dynamics, or the inverse problem of kinematics, right. So, I know the end point. Suppose I have to reach that end point, I have multiple effectors, right, multiple links with different varying ranges of movements, right. I can do that. I also have movements at the shoulder. So, when I have to reach that point, what could be the joint angles that would or the set of all joint angles that would help me to reach that point, right. That is the inverse problem. Or if I know the configuration of the chain, or if I know the individual joint angles, I would like to know what is the end point that could be reached. If I know the individual joint angles, I should also know the link lengths. Suppose I know both, then I could predict what would be the end point that would be reached. That is the forward model, right. So, it turns out that the cerebellum implements these, or the internal models in some sense, or it is believed that these internal models are implemented in the cerebellum to account for the dynamics. For example, right, if I am making a movement, here is an example that is shown of a person trying to make movement of the forearm. So, that is the forearm, I am trying to make that movement. In healthy individuals, right, when they make movement of the forearm, the shoulder does not move. Why? This is because it turns out that in healthy individuals, normal as in healthy or typical individuals, right. It turns out that these individuals account for what are called as interaction torques. So, when I move a more distal part of the body, it also causes movements of the more proximal region. For example, when I am moving the forearm, the movement generated by the forearm also produces unintended torques or unwanted, or at least interaction torques, because this is connected to the proximal part of the body, this is connected to the upper arm. Because of that reason, it produces an interaction torque on the upper arm, causing the upper arm to move, yet in healthy individuals the upper arm does not move. Why? Or there is no movement at the shoulder joint. Why? Because the cerebellum anticipates that this movement of the forearm could in some sense cause movement of the upper arm, and appropriately sends muscle or commands to the muscle, not directly, indirectly sends commands to the muscle, so that movements that negate or muscle activity that negates movements that could be produced here are produced. So, simultaneous or properly timed sequence of movements or properly timed sequence of muscle contractions are produced at the shoulder joint to prevent any unwanted movements that could happen. This is happening in the healthy people. But when a person has cerebellar damage and the same movement is performed by this person, usually you see there is some unwanted movement. So, that means the system is not able to account for the possible effects of dynamics, for the possible interaction torques, right. So, there is this failure to compensate for interaction torques. So, also we said that the cerebellum anticipates movement. Suppose what happens if this ability is compromised? This has been tested in animal models by cooling of the deep cerebellar nuclei, or which is a temporary perturbation. When that happens, what is observed is that there is a delay due to cooling or inactivation of neurons in the deep nucleus. Why does this happen? So, delay of what? Delay of antagonist movements or delay of the onset of movements. So, what happens is, we know that in healthy individuals to perform fast reaching actions, we discussed early on that there is going to be a triphasic EMG pattern. So, that means agonist movements followed by antagonist movements. For you to come to a smooth stop, the antagonist activity must start relatively early in the movement, so that you are able to stop at the point where you want to stop and in a smooth manner. So, when I am performing that, when I am performing the extension movement, right. So, when I am performing that movement. So, the extensor in this case is the triceps, but for me to come to a stop at the point where I want to come to a stop, suppose that is the point where I want to come to a stop there. If I want to come to a stop, appropriately timed activity of the biceps is required, so I can stop smoothly, right. Now, that requires sequencing of these two activities, that require timing of these two activities. In healthy individuals and in healthy animals, this is relatively normal, and people are able to, and people and these animals are actually able to properly time these two things. So, agonist activity which is followed by antagonist activity, and this antagonist activity starts early on in the movement, so that movement comes to a relatively smooth stop, and that is also relatively accurate. So, this is in some form of feed forward or anticipatory control, right, anticipatory modulation. Why? I only want to stop the movement when I want to stop. In that sense, why do I have to activate the antagonist so early in the movement? Actually, it starts at about 20-30% into the movement cycle, right, not on the second half of the movement. So, that means I am expecting, I am anticipating that I will have to stop at a particular point and appropriately timing the activation of the antagonist. This is found in intact healthy typical individuals, right. When you cool the deep cerebellar nuclei, or when you inactivate temporarily the deep cerebellar nuclei, this ability is compromised. Then what happens is that people slow down. One is that there is slowness of both agonist and antagonist, but also this ability, this feed forward feature of this movement is compromised to such an extent that the only control or the only modulation that happens is purely feedback, then the movement is dysmetric. So, the movement resembles dysmetria, there is an overshoot or the antagonist activity does not start early enough that, or it starts so late that it cannot stop the movement until after there is an overshoot. This is typical of cerebellar damage, dysmetria. There is either an undershoot or an overshoot in dysmetria, but this is a classical case, typical case of cerebellar damage when the person is not able to meet a target, they are overshooting, dysmetria, right. So, that shows once again the important role or the crucial role of cerebellum in regulating feed forward or anticipatory actions, right. Now, we will proceed to discuss the possible role of parallel fibers in motor coordination. What could be the role of parallel fibers? We have 100 billion of these. What could be the role? It turns out that recording from Purkinje cells during action, during activity in live behaving animals, and by selective cooling, by selectively perturbing the system, has taught us a few lessons about what could be the possible role of these systems. It turns out, we discussed this earlier in one of the previous classes, we have discussed that the Purkinje system is thick in the anterior posterior direction and very thin in the mediolateral direction. I said that, suppose this is the paper, it is going to be thin in this direction, but you are going to see a breadth in that direction. We saw that in one of the previous classes, and it turns out that the parallel fibers run perpendicular to the Purkinje fibers, is it not, Purkinje cells, that is what you are seeing here. So, here is the tree, this is a 2D structure in this, so the third dimension is not shown. So, this is the Purkinje cells dendritic tree, and what you are seeing here are all the parallel fibers, right. So, what has been shown is that there is synchronous activity of Purkinje cells. So, there is also, we have also discussed that there is somatotopy of neurons in the deep nuclei. I mentioned that that there is a homunculus that is upside down and turned sideways. We discussed that or in other words, the anterior side is the tail or on the posterior side is the head, right. We discussed this, and also there is synchronicity of activity of the Purkinje cells in the mediolateral direction. So, that means what? That means in the mediolateral direction there is representation for different muscles. This representation is there not in the cerebellar cortex, not in the molecular layer, but in the deep nuclei, and they project somatotopically. So, they receive information from the Purkinje system somatotopically, this is what we have said. So, there is topographic representation in the deep nuclei. So, when Purkinje cells are active synchronously in the mediolateral direction, that probably means there is an attempt to coordinate muscles at multiple levels, and this is true, this is what we just discussed in the previous slide, right. So, here what is happening is that this movement or change in this joint angle from about 90 degrees to an acute angle is due to different muscles, is due to biceps and triceps, whereas that movement of the shoulder is due to different muscles, right. Here we do not want this movement. So, there is coordinated timed activity of these muscles in healthy individuals, so that only this joint angle change is visible, is manifest, whereas any other there is absolutely no change in the shoulder joint angle, in that joint angle, right, that remains practically the same between the two configurations, right. So, that remains the same in both the configurations, that is what is shown, right, that is in the healthy individual. So, that means that probably the parallel fibers activate Purkinje cells in such a way that there is synchronous activity of Purkinje cells or there is appropriately timed activity of Purkinje cells that leads to multi-muscle coordination, right, and this is critical for movement control and movement modulation. So, what we have seen in today's class is that there are 3 organizational principles associated with the regulation of movements by spinocerebellum, and what are these? These are what are these? These are that there is Purkinje cell and deep nuclei activity in relation to movement, and there is anticipatory or feed forward activity in the cerebellum, and then probably and more controversially there is this so called internal models that probably encode dynamics, believed to encode dynamics of movements. Yes. And we also discussed what could be the possible role of parallel fibers. The parallel fibers synchronously activate Purkinje cells at multiple regions, resulting to multi-muscle coordination or motor coordination. So, this is a crucial function of the cerebellum. So, cerebellum does not control movements, but rather regulates, modulates, coordinates movements, which is critical for everyday function. So, we will continue this discussion in the next class. So, for now we will stop. Thank you very much.